That's easy. So Nicola, okay.
Lovely. So I'm just going to be talking today about monitoring of biodiversity with environmental DNA. Next slide, please. Thanks, Mars. Shall I just sort of put my hand up so I don't have to keep saying next slide? So I'm going to go over what it is, how you extract it from a sample, why it's useful, how we're using it in Herts Middlesex Wildlife Trust. We've got two examples. We've got one of a sort of multi-species use and then one of a single species at the end, and then I'll just go through future applications. So DNA is all around us. As you're sitting, as you're walking, it's in the air, the soil, and the water. We can collect that with an environmental sample, and that will help us understand the species that are found in that environment. So this is actually a revolutionary technique. It's sort of been around since about 2008, and it gives us the ability to understand biodiversity across different habitats, and most importantly, it's in a non-intrusive way. So DNA is made up of different genes, and each gene has a distinct pattern of nucleotides using the A, T, C, and G bases. So when we go through profiling of a species, you see these lovely color charts, which is probably what Miles was referring to earlier about the art applications. And we generally use the CO1 gene, or part of it, and that's best for barcoding animals. This includes macroinvertebrates, fish, mammals, and birds. And this gene is identical, well, almost identical between members of a species, but relatively different between different species. So as you can see here, the top two are both the same species of Astrapis butterfly, but DNA profiling has uncovered cryptospecies. So actually, there are 10 different species, cryptic species. There's 10 different cryptic species discovered through DNA barcoding. And if you look at the colours, I've pointed out the areas where they're actually different there with the red circles. But if you look at those two, they're very similar aside from that, almost identical. But the bottom two are different species of owl. And as you can see, the two different species of owl are quite different, and they're in turn quite different from the species of butterfly. Okay, so next slide. So we can quickly discover species within a habitat. It's low cost compared to multiple taxonomists, but it depends on complete libraries, such as BOLD, to identify those sequences once they're identified. So this is BOLD, and this is a library collection of species. And what's great about this is we have a taxonomic, an individual species specimen for each species. And along with that goes the barcode. So that is held in a reference library, which scientists and future investigations can go back and look at that and just check morphological features against it. And it's a really useful reference. Whereas in the past, we may have just had the DNA reference, but with no actual species individual attached to that. And that has led to some issues with the databases in that we don't know, we can't be 100% certain that they are correctly identified. And that does sometimes show up in our results when we're looking for these species at the end. So some of these cryptic species, I meant to say, sorry, they could be genetically isolated populations. And it does highlight a hidden diversity that taxonomy hasn't shown us before, which is really quite exciting. But obviously it brings with it its own problems. We also get to see the geographic location of the species in BOLD. And as you can see on the map, that's where the species have been recorded, where it's highlighted in orange and yellow. And it tells us there's been 37,000 sequences for this individual species and who's contributed to that as well. So, so what they tend to do is take a part of the species that's not used for morphological identification, such as a leg, and that's what's sent off for DNA profiling, which is used in this reference. And that's really important because these records are now getting complete, and then we can trust the results, and that's what we need to do with DNA. Okay, next slide, please. Next slide, please, Miles. Thank you. So this is how we've collected and processed our samples for our water testing. We've collected water samples and filtered them through some filter paper. The filter paper is collecting the organic matter which will have the DNA attached to it. Now, you could also do this from air or soil. With soil, you just take the soil sediment sample back to the lab. With air, you use what's like the back of a hairdryer that sucks the air through a filter paper, and you might leave that out for about an hour or so. And that filter is then taken back to the lab, and the DNA is extracted in a similar way to that for the water sample. So the DNA is extracted from the sample, it's purified, and then we use primers to amplify the target gene. So it goes through PCR, which is polymer chain reaction, and we can either look for a group, as in multi-species, or we can look for a single species. Now, if you're looking for a single species, you can amplify, you're just amplifying the DNA from that particular species, so it's a lot more, sorry, I've got a word like delicate. That signal from the species may get lost if you're doing a multi-species analysis. But if you're looking for something that maybe isn't there in high numbers or quite elusive, it's quite good to do a single species analysis for that reason. We then send that for sequencing. We get back that lovely barcode, and we run that through the library, which such like vole that I've just talked about. Once it's run through the library, you then get a percentage match, and that will tell you what the closest samples are in that reference library, and you can see how confident you can be in your results. So obviously we're looking for 100% or high 90s. Your DNA strand that's been multiplied might not be in 100% condition when it's taken from the environment, because it might start to break down. So we're looking for very high percentage matches. Also, we can't currently look at abundance, although that is a developing field. It's quite difficult. You can look at read counts, and we can look at relative abundance of read counts, but different species will emit different amounts of DNA into the environment. So it really only works if you're looking at single species. Slide, please. Okay, so as I touched on before, once the DNA is in the environment, it can start to break down. There's different ideas on how long it can persist, but it will start to degrade, especially with high temperatures, high pH, and sunlight. Also, when you're thinking about a river system, the DNA is in movement, it can move downstream, so you've got to take that into consideration. If you're looking at something like a lake, we've obviously got stratification of where you might find the DNA, but you know that if you're getting it from that lake, it's most likely from that water body at that location or somewhere in that location, so it's a bit easier. So when you're looking at your results, you need to think about how it got to that location. It could be coming from downstream. It could be the contents of another animal's digestive tract. It could have blown there in the wind, or it could be getting to the site through people. It could be on your equipment, or it could be brought there from dog walkers or equipment that you've used at a previous site being transferred to a new site. Okay, next slide, please. Okay, so how have we used environmental DNA at Herts Middlesex? Our main study, which happened in started in October 2024, we worked on a project with Natural History Museum, which also involved the Chilterns as well. This was called Gene Flows. Prior to that, the Natural History Museum had done a project called Gene Pools, and this is where they asked citizen scientists to sample their ponds, and they took those samples back, they processed them, and they made a lovely website about that. And then they had some funding, luckily for us, to trial that method on rivers. They again wanted to use citizen scientists. So we got involved with them to look at the total biodiversity across some of our rivers. We used, the questions we had were, can we use the citizen scientists to collect the samples on the rivers? Can we use to look for species of interest? So obviously we're quite big on water voles. We also really love a stonefly, especially up in the top reaches of the rivers. But also, how effective is it looking for invasive species? Additionally, we had some other questions. We had a lot of questions, and in hindsight, we had too many questions, but we are working on it, and it's still, the data set is there, and we will be working on it for a very long time. So another thing we wanted to look at was the impact of sewage treatment works and barriers on our species, and that is stuff that we will come to in future. Yeah, so I'm also working on a single species DNA project currently, looking for our native eel, and that is in the lower Lea catchment. Next slide, please. Okay, so this shows for the gene flow project where we decided to sample. The triangles are barriers as they are in the barrier app. They're not all 100% sort of confirmed. The yellow dots are riverfly sites. So we thought across the rivers, we're going to try and do our samples at riverfly sites because we've got people that know the rivers there, so we can come back and ask them questions, and we've got some of their data. And the red sites are additional ones that we wanted to pop in at the top and the bottom, mostly of our rivers, to get that additional data. And as you can see on the, I think that might be the store. No, it's not. Which one is that? The far right anyway, on the Lea catchment. We wanted to know what was going up in the top of that catchment, because obviously there's no riverfly happening there either. So in addition to the environment DNA samples that we took, we also took a preserved kick sample at our top and our bottom site from each of these rivers. And the aim is to then compare our taxonomy list with our environmental DNA list for each river to see how they match up. Now, we took 21 samples. I'm not sure where the extra one came from, but there's 21. We've got 5 left to do, and we're still cracking on that through that. Myself, Louise at the Natural History Museum, and also a volunteer. So we're hoping that we can use that in the future when we've managed to get through that. Next slide, please. So from all of those environmental DNA samples, we found 412 different families and a whopping 1,897 species, which was really quite cool. And we also found not just riverine species, we found terrestrial species as well, which was quite exciting. So yeah, next slide, please. Now, the Natural History Museum, the Natural History Museum has created a really nice data dashboard where anyone can go and investigate our results. And you can go down to site, you can go down to river, you can get an overall analysis of your river, you can look at your site, and you can get the 33 species of extended riverfly as well are highlighted. So you can use that if you are a riverfly volunteer as a basis to see what can you see at your riverfly site and what have we found. But across this, we did find 5 species of conservation concern. So we found the eel, European eel, the water vole, Eurasian otter, water shrew, and the great crested newt. So that was one of our objectives. We've also found invasive species, demon shrimp, the signal crayfish, and the one that I can't pronounce. So unfortunately, the signal crayfish was very well represented, but we did know that already. But the demon shrimp was only found at one site, and that's the only site we're aware of it being at. So that is actually quite good news in that we could use it as a sort of early warning system for our other rivers, just to see where it's spreading. So it is picking it up even at a multi-species level, which is great. We only found 2 records of eel, which is a critically endangered species, and that's what I'm working on in another project with a single species assay. So hopefully we'll have more luck with that. Next slide, please. So the website that's been made gives a nice taxonomic composition of all the species that we've found, and you can get that for each river as well. The majority are actually aquatic insects, so that is quite good, but we did obviously find a host of other terrestrial species as well, and water-based species. Next slide, please. So we're currently in the process of trying to understand the data. What's been really great about this project, although my time at the beginning was actually through the Casco project, and Louise was funded for 9 months through the Natural History Museum grant, it's currently now all been done under voluntary effort, basically. So myself and Louise are working through the data, and also a volunteer called Colette is helping us sort the samples. And the citizen scientists themselves on the rivers have been feeding back what they know about from their sites. So it's been a really great example of sort of people power, I suppose. Yeah, next slide, please. Just take some water. So how are we using the information and what is it telling us? So on the Elwenbrook, I've been informed that the stonefly, the picket stonefly, has only been found at the topmost site, and those dots are sites, riverfly sites that we've sampled. So those blue dots we sampled out, and the grey dot is an old riverfly site. So we found the picket stonefly DNA at the top site where it is found in manual observations. It was also found in the next site just downstream, but after that it wasn't found. So this is showing us how far this DNA is traveling. It's not found in manual observations at the second site, but at that second site there are watercress beds. Now, watercress beds, as you know, they're going to slow the water down. There's a lot of vegetation there, so any DNA is likely to sediment out. So this is a reason why it's not moving downstream. So it's showing us that actually understanding the environment can help us understand how far the DNA may travel. And it's not just a case of, oh, I've got my eDNA results, this is exactly where it is. We need the context of local knowledge to put that into perspective and to be useful as well. Yeah, so next slide, please. So here I was looking at the river bean at the total ESV reads. So that's the total strands of DNA that have been amplified for all fish at all the sites that we looked at. And I was really expecting a straightforward sort of zooming upwards curve of, you know, the DNA is increasing as we go down the river because it's picking up DNA as it goes. But what I found was something quite interesting. It's actually showing me that DNA must be dropping in and out, dropping out and then increasing at points of high fish activity. So this graph actually shows us the Lammas where it dips down. It's a really slow moving, compounded section of river, very sediment heavy. And yeah, there's not really much movement there at all. Then you get this big spike on the graph, and actually that's showing at the same point as where there's a large lake at the Woodhorse State, and there will be many fish in that lake. So that's a big injection of DNA. So this is telling me that if you just looked at total ESV reads, you could start to think about the conditions in your river and where you might find larger amounts of fish, and where you might find slower moving sections of your river as well, which is quite interesting, I thought. Next slide, please. Then I looked at species of fish at each site. So again, we're going from left to right, we're going downstream. I looked at the species of fish present, and as you can see, it changes a bit. We do have goldfish, and I did check that with the EA. Apparently there are goldfish there, so that's not, although I thought that was a mistake in our results. No, they probably are there. So they're pretty similar to the EA data for fish, and what we'd expect, although the trout is there a bit, which I was thinking is quite unusual. That could potentially be moved upstream on people's equipment or their feces. One thing that did show up in this was that the bullhead, which we all know is quite abundant, it wasn't showing up in our results, but actually on investigating our library, it turned out that our native species wasn't there, but a very similar relative from Europe was there, and that's what was showing up in our results. So we know that it's been misidentified through the library. So we're quite confident that that is our native bullhead species. So that has been added in. And also we had one, the Rotillus, I cannot pronounce italic, sorry, Latin names, Rotillus frizili, which it was is probably a closely related species, but it's not actually a species that we have on our river. So on the whole, it's giving us a good overview, but we do need to interpret it with other knowledge. Yeah, next slide, please. Yeah, so the chess, the chess, Paul Jennings and the chess rangers helped me out with the list of species they know are there. And there were a few that they found were missing. We know that the bullhead is there because they had the other record, but the perch and grayling didn't show up, although they have showed up on other records. So that's a bit of a strange one. It could be that it wasn't in the water of the sample that was taken and that they were somewhere else, or it could be that perhaps this is an example of where you want to do multiple sampling to cover so that you get a wider sample, basically. So you might take 3, which is what you would normally do in a scientific study. So a triplicate. That might help show that up. But it is interesting that it's not picking everything up, but it did pick up quite a fair few of the species there. Next slide, please. So what I thought was really fun was all the different species that showed up on the River Ash. So we've got things like red kites, and we've got badgers, which are my favourite. We've also got millipedes and many beetles, and okay, thank you, mice, water shrews. There's so many species here that it's showing us that through monitoring the water, you can actually think about the wider environment as well and the wider habitat, which I think is really exciting. And it also picked up some things like horse and sheep and cow, which are likely to be in the air, dog and pig, but then wolf and bonobo. So we're not quite sure where the bonobo lives. It's possible someone has one, or it's come from the zoo, or it could just be a misidentification. But also wolf, we think, is probably some ancient dog DNA, most likely. But on the whole, most of the species are ones we would expect. So there's not that many that we wouldn't expect. Yeah, so next slide, please. Yeah, so Steve Brooks has done some his own sampling of preserved samples for another project we were doing at the same site. And he noticed that not all of the caddisfly were showing up in the eDNA that were showing up in the manual sampling. And we're not quite sure why this is. And a lot of them were the, which ones were they? I can't remember. Caseless caddis, that's it. The caseless caddis were not showing up as much as the case caddis, and we really don't know why. Yeah, that's something we're trying to figure out. So that's a question that's thrown up. Next slide, please. The water voles. So we wanted to know, would it help us to understand where water voles were and where they weren't? So I compared them to the manual surveys, and 2/3 of the time we have the correct correlation, but they were showing up higher up in the river system than we expected them to be. So this is showing us everywhere we thought they were, they're showing up as positive, so that's great. But it did show up in a couple of other locations, which I need to check, have been checked, and I need to find out if they found them there or not. But it's showing us that we could potentially use this for water vole and then go back and check those sites that where we're getting positive results to see if there are signs there. Next slide, please. So another way of looking at it, on the river rib, the top and bottom sites here, showing us with our results, the top site and the bottom site, because we can look at the BMWP score and the ASPT scores, and also the conservation species. We're getting the conservation species and the invasive species at the high site. And this site is going through fields, it's got trees, it's got a reasonable habitat. The Buntingford site is our lowest site, it's poor, it's got low scores. And actually that site is running through the town, it's completely canonized, it's very flashy, very low water, and the habitat is just not there. So we can use the overall results to give us a little picture without even visiting the site. Next slide, please. How much time have I got, Miles?
Probably about another 5 minutes or so.
Okay, I'll try and be quick. On the MinRAM, the top site for when we look at the extended army scores, and we are showing us this is our top site for eDNA. Also, we have Simon Stebbings, who's doing a lot of extended riverfly there. It's coming out as the top site there on the MinRAM, this Turenbury site, through his manual studies. So it's showing us this really good correlation with the extended riverfly is actually agreeing with our best site for eDNA. So that is actually really quite interesting. So it's showing us that we could potentially use this at sites that we're not doing extended riverfly to have a good idea of what could be there. But as we can see, there's a couple of caddis missing there, which we need to work out why they're not being picked up. And next slide, please. I remember how many slides? Yeah, so almost at the end now. How accurate are the results? So I've gone over a few issues. Positive is we know the invasive species, the shrimp were showing up only where we knew they were, and that the water voles, we were getting positive results where we knew they were. But there are some species that aren't showing up when we know they're there. Flatworm is another one that wasn't showing up. And some species are incorrectly identified, as I suggested with the bullhead and the wool. Next slide, please. But something really exciting came up. I was listening to another talk about virozoans. I went back and checked our results, and we've got virozoans. We did actually find them in our preserve samples as well. But this was a species we didn't know was there. It did actually come out through the barcode through the library as an incorrect species because they're not very well represented. But it is showing us that we can use it to identify species that may be in your river that you could then go back and follow up with taxonomist experts. Next slide, please. Okay, so let's go to the next slide. I'll do this bit at the end. Yeah, thank you to our partners. Next slide. Just quickly go over the IL survey and what we're doing. So this is going to be a single species survey. We're going to, because we're looking at a lake, we're going to take samples from around the lake every 25 meters. We're going to pull those together and take 10 overall samples. So you'll take the water sample from a couple of locations, 2 or 3, mix it up in a bag, and then filter that sample. Yeah, so it'll be very exciting to see if we can find the eel, our native eel, in this lake. It has, the reason I picked this one is because we had manual surveys showing up to about 2013 that they have been here, and eels are quite elusive. And according to the EA fisheries officer, sometimes they don't come up in the electrofishing because they're quite heavy and they can sort of get themselves in nooks and crannies. So it could be, we want to find out if they're there, and this is a pilot project, and if this works, we'll be spreading that out along that Lea Valley site. Yeah, next slide, please. Okay, so I will just quickly go over that other slide. So how can we use the data? So it's showing invasive, it's okay, you don't need to go back, Miles. It's showing invasive species, it's showing species of interest, it's highlighting places you might want to go back and do manual observation. It's great for overall biodiversity monitoring. As we saw, we're getting terrestrial species as well. It's great for biodiversity ranking of your sites, but we need the context of local knowledge. You need to use it as part of a bigger picture and in combination with the other methods. it is improving all the time, and with improved libraries and more studies, this is only going to get better. It's also great that we've got this reference collection, and we can go back, and in the future, once the libraries are improved, we can run our results through that. And as I said, with the biozoans, I did go back and check, oh, do we have them? Yes, we do. So that data set is there to be used now. It's also open source. And yeah, in the future, hopefully we'll be able to start looking at abundance. So that's me done. Thank you for listening. There's a couple of good resources there.
Brilliant. Thank you so much, Nicola. That was really interesting. Right, has anybody got any questions for Nicola at all on that and what they've just heard? Everyone put their hand up. Oh, Adrian?
I'm kind of going, we're involved in gene flow ourselves on the chest. We did the handle measurements and things. So actually we found the data really useful. I'm interested in when do you think we should perhaps rerun this, this data set compared to, obviously we do citizen science on these rivers, we're making changes. What's the frequency of doing an eDNA sample, do you think?
Well, I mean, it's changing all the time. I imagine, well, if you've done any kind of habitat changes or there's been any changes to the river, I guess that's what you're looking for, really. Because if you're thinking of it as a total biodiversity survey, it's what's changed. So are we going to repeat it in a few years, thinking about the temperature of the rivers? Or if you've done a big amount of improvement works to the rivers, has that changed? So I would think of it as a kind of similar to any survey work that you would do, really. We really want to run it again, so we are looking for some funding to do that, but we want to get through these kick samples first, so that we can start comparing the DNA with the kick samples as well. I think next time we might not do the kick samples, so that we can have some more time to understand, actually delve into the data and what it's telling us. Yeah, so I think it's, what is your question you're trying to answer would be how long you then do it again. I mean, ideally, I'd do it every year just to see the changes. But again, thinking about it with surveying, it depends on the time of year. And we did this sort of October because of when the funding came through and when when Louise was available for lab work, we had to do it at that part of the year. But actually, if I was to do it again, I'd think about doing it springtime, just to get that different sort of suite of species that maybe wouldn't be picked up in the October. So that would be something I would think about, I guess.
It's quite interesting correlating that with the presence and absence of water voles, because we don't do water vole surveys on every river. Where we don't do a water vole survey, it might be quite good to get a sense of where the population is, not how big it is, but where it is on the river. I think that's quite interesting, sort of a gap filling, if you like. But yeah, there's a cost involved, isn't there? Which we all have to be a bit aware of.
Yes. So the single species is coming in about the eDNA that I'm about to do. It was about 110 plus VAT for citizen science for one species. I think going up to, it was like 140 plus VAT if you want to do on the qPCR, which gives you sort of a little bit more inkling on abundance. And then I think it was around 250 plus VAT if you want to do like total biodiversity, basically. Yeah, per sample. So it's expensive, but not as expensive as taxonomists. And yeah, and I think with the voles, so one of the many things we want to do with this data is look at all, because we've got quite a lot of water vole survey data, manual surveys. So I want to actually compare that with our eDNA completely and get some stats out of it. So I'd only managed to do the bean because obviously this is now more of a pet project than an actual project. So I don't have that much time to do it. But yeah, there's so much out there, and for students as well to use it to investigate our data. That would be great.
Yeah. Okay, great. Thank you. Any further questions still for Nicola? I've just got one then, just briefly, just around citizen science, really. Who was collecting this data for you? Were you getting citizen scientists out there doing it, or was it yourselves doing it?
Yeah, so so I was involved in Casco on the River Bean, so we had quite a lot of volunteer involvement on the Bean already, And then we reached out to the other riverfly coordinators and other people working already that we knew, the contacts on those other rivers, to get involved. And the same in the Chilterns. We got Hannah, who was there at the time, to pass it on to their citizen science coordinator to get people involved. So yeah, we were trying to get people involved. We didn't have much time to do any extra outreach, but the people that we already knew, we were sort of getting them involved. Yeah,
that's great. And how do you present the results to them? Have they seen those results yet? Or?
Yeah, so I did a presentation, and Louise did one at our Riverfly event. We have normally a yearly Riverfly conference combined with the Chilterns. So he did a presentation there. They've also got the website where they can go in and investigate, and I'm happy to take any questions from anyone on the rivers, and trying to look into the questions they have and the data that they are providing in addition to that as well. So we've tried to sort of present it back to them, because I think it's, and it also, we're still in that conversation with people, and what have you found, and take our eDNA list and see, can you find those species at your site? So there's still a lot more to be done with that we can, in the future. But I think even just thinking of it as we've taken this reference now, so for the future we can look back and say, oh, what did we find at that time? Because we have got the sequences are there. We might not know what some of the species are, they might be incorrectly identified, but as the libraries improve, we can go back and set that back through the new improved libraries. So that's still available. Yeah.
Okay, great. Thank you very much. Any further questions? Anybody? No? Okay, brilliant.
It's okay.
Thank you so much, Nicola. That was really, really, really useful and really, really interesting. As I mentioned at the beginning, we'll put the presentations will be put up on our website, and we'll share the slides and everything with you as well. So yes, brilliant. Well, thanks so much, everybody, for your time today. Really appreciate it. Massive thanks, Nicola, for taking the time to put all that together and to come present it. Very much appreciated by everyone, I'm sure. So thank you so much. And yeah,
Thank you.
If you all enjoyed your lunch, or will enjoy the lunch you're going to now. So cheers, everybody.
Thank you.
So much.
Thanks, Miles.
Bye-bye.